Hello, my AP Biology minions. Today we're going to discuss translation and how we get from that wonderful transcribed and edited messenger RNA to the proteins, polypeptide chains, and enzymes that you all know and love. First, on this journey to understand how your cellular machinery is able to translate, we need to talk about ribosomes and what they are and their structural components. Okay, ribosomes are a cellular organelle. All right, and they are where protein synthesis happens. That's probably something that you've heard many a time before. Now, what that means in terms of where protein synthesis happens, it means that ribosomes catalyze complementary binding of transfer RNA anticodons to messenger RNA codons. All right, so that whole complementary thing, A equals U, C equals G, okay, that complementary binding is what allows transfer RNA to attach to messenger RNA in the appropriate sequence. So the ribosome catalyzes that. It also catalyzes the forming of a peptide bond between amino acids on tRNA molecules. All right, and peptide bonds are the bonds that are used to form polypeptide chains, which become structural proteins and enzymatic proteins. Ribosomes have two main structural components. They are small and large subunits. You can see in this diagram here, the red is the large subunit and the blue is the small subunit. Those subunits are made of protein components and ribosomal RNA. It appears from recent structural analysis of ribosomes that the protein components tend to make the outer structural aspect of it, and the ribosomal RNA seems to be located mostly in the places where the messenger RNA and the transfer RNA interact. Further evidence of the sort of ribozyme component of ribosomal RNA. This structure right here, just as a reminder, is transfer RNA, and what you'll note, all right, is that this right here is a sort of chain-like representation of it, and what you have right here is the actual like three-dimensional model of the messenger RNA or transfer RNA single strand itself. This structure right here is where the amino acid binds, and so this structure is slightly different depending on the amino acid that's meant to bind there. Down here, okay, you can see the anticodon, all right, and one of the things that you'll note most interesting is that transfer RNA starts at 5 prime, and that means that it ends here at 3 prime, which means that the anticodons go like this, which is 5 to 3. So does the messenger RNA. So it's one of the few times you'll see complementary binding exist in situations where there is not a antiparallel strand. Okay. Inside these ribosomal subunits, there's 3 locations, and we'll see those more specifically as we move forward. The first one is known as the A site. It stands for aminoacyl tRNA site. The second site is known as the P site. It's the peptidyl tRNA site. And the final site, the E site, is the exit site. Again, we'll get into this, but at the aminoacyl tRNA site, that's where the appropriate tRNA is brought into the ribosome so that it can be used for polypeptide chain forming. The peptidyl tRNA site is actually where the formation of the polypeptide chain occurs, and the exit site is when the now amino acid-less tRNA is removed so it can go find a new amino acid. All right, so let's talk a little bit about what goes on in the process of building a peptide. First thing, the messenger RNA is generated in the nucleus, and that's important. Transcription happens in the nucleus, as does the editing. You know, we talked about how you go from primary transcripts and pre-mRNA to this whole messenger RNA thing. All of that stuff happens in the nucleus. And then remember, the poly A tail is used to extract the messenger RNA from the nucleus. And once that happens, that messenger RNA ends up sandwiched between ribosomal subunits. All right. Now, tRNA is then brought in from surrounding fluid. Depending on the type of cell, this surrounding fluid could be cytoplasm, it could be fluid, cellular fluid in the endoplasmic reticulum. There's a lot of different options there, but that tRNA is brought in from surrounding fluid into this ribosomal complex. All right. Important aspect of this for the means of efficiency. This can happen with multiple ribosomal complex on one strand of mRNA, so they can actually be making multiple polypeptides from the same messenger RNA at once. And when this happens, we call it a polyribosome. And that just means that there's multiple ribosomes reading that messenger RNA, translating that messenger RNA at once. Now, this whole process of building a polypeptide inside a ribosome has three main stages, all right? The first one is initiation, we start the process. Translocation, we move the ribosome down the messenger RNA. And then termination, where the polypeptide is fully formed, so we need the process to stop. So let's talk about each one in turn. Initiation. All right, so the first thing, every polypeptide begins with the amino acid methionine. So the very first part here is to get a tRNA that's carrying methionine to bond to the methionine codon on the messenger RNA strand. Now, some polypeptides, protein initiation factors, combined with an energy molecule known as GTP. It stands for guanosine triphosphate. All right, and what happens is enzymes phosphorylize the GTP, breaks bonds, which releases energy, and that actually brings in the small and large subunit together. So let's think about this in terms of order. Messenger RNA binds to small subunit, all right? Then the tRNA with the methionine is brought in. This causes protein initiation factors and an energy molecule to bring the ribosome complex together. And that forms what we call the translation initiation complex, okay? Now, the tRNA, when it's set in here, is fit into structurally what we call the P site on the ribosomal complex, all right? And then a number of things happen, all right? First off, we have what's called codon recognition. So, again, polypeptides, what they do is they start to bring in the next appropriate tRNA to the A site, all right? And then complementary binding occurs at that A site. Energy is released, and then what happens is, as that energy is released, ribosomal RNA inside the large subunit catalyzes. Remember, it allows the reaction to happen at a greater pace. It catalyzes the formation of a polypeptide bond between the two amino acids. The one that is on the tRNA in the P site and the one that is on the tRNA in the A site. And the forming of that bond requires energy, okay? And that's what I just said. The next step in this process is called translocation. Think about it, it makes sense. We start the process. Then, once we've got our first bond, well, the ribosome has to move down the mRNA strand so it can keep reading it. And that's what translocation is. It's moving this whole complex down the messenger RNA strand so it can continue to be read appropriately. So in this process of translocation, the transfer RNA that was in the P site now moves to the E site and leaves, and that tRNA no longer has its amino acid attached. The tRNA that was in the A site now moves to the P site. And this one doesn't just have its amino acid attached, it now has two amino acids attached, okay? And that is the building polypeptide chain. Now, this process, again, requires energy. Hopefully what you're getting out of this is that a lot of energy is required to complete the cellular processes you rely on for survival, all right? And then, again, a new tRNA is brought into the A site, and then this whole thing can just repeat. And this continues to repeat until you get to the point where you hit a stop codon. And when the stop codon hits the A site, all right, release factors, again, polypeptides, other chemicals, hit the stop codon. And then what they do is they do what we call hydrolyze, and that means that they break the bond between the growing polypeptide chain and the tRNA molecule it's currently attached to. So that way now the polypeptide chain is free as a bird, to live its life as it sees fit. Then, once that happens, the whole complex separates and falls apart until something causes those pieces to be used for another initiation. Okay? I know, it's complicated, and it's a lot of words, but fortunately for you, as a trained educator, now I have pictures for you. All right, so let's take a look about this, all right? Right here, you've got A site, P site, And then here, even though it's not so obvious, is what we call the E site. So basically what goes on, I'm going to leave the strong terminology for the previous slide. Basically what goes on is a amino acid gets, or I mean a tRNA holding amino acid gets dragged into here. And then GTP molecules, enzymatic, or not enzymatic, but protein factors that allow this process to occur, drag this large subunit and this small subunit until they sandwich this right here, messenger RNA, and this tRNA inside this big complex. Energy is released, and then a new tRNA is brought into the A site. More energy is released, and this amino acid is attached to this amino acid. More energy is released. This, with the growing amino acid chain, now moves from the A site to the P site. And another one comes in, this one leaves, and the process just continues to happen until a stop codon is reached. And then the whole thing falls apart. Energy is released at almost every step. The energy that's released comes in the form of GTP, guanosine triphosphate. All right? But the most important thing here is, in the P site, you've got the location of the elongated amino acid chain. And every time energy is released, the chain is moved from the P site to the amino acid on the A site tRNA. Then the A site tRNA has the entire chain, and it gets moved to the P site. And then a new one comes in the A site, and the one that was in the P site that no longer has an amino acid is now left out. It goes and finds a new amino acid. And you can see over here, this 2 tRNA just free floating in the cytoplasm. All right, or in the case of many eukaryotic cells, inside the endoplasmic reticulum. Okay? So, if you really want to figure out how to think about this, take a look at this picture and the previous slide and just sort of go back and forth. All right? Now, you'd think after translation was done that, hey, we've got a polypeptide. Unfortunately, nothing in biology is ever that simple. So, post-translation, polypeptides are what we would call the primary level of protein structure. Polypeptide chains need to be folded into other levels of polypeptide structure, secondary, tertiary. And that folding gives the polypeptide a globular shape, and then that shape is its structure. And you can never forget, structure equals function. And so it's actually this folding of this sequence that gives the polypeptide its structure. Now, a lot of this folding is determined by the chemical nature of the different amino acids. So, in fact, the order of the amino acids is incredibly important to that structure. But the folding itself is done by something called chaperone proteins. And you can see this right here. All right. Chaperones. They're more than just what keeps you from making out at the dance. All right. You see up here, the blue and the green is the new polypeptide chain that's being folded. So all of this right here. And the red is the chaperone protein that's aiding it to make sure that it folds into the appropriate structure. There's actually a big problem that happens if these proteins aren't folded properly. They create a structure that's called a prion, which is a misfolded protein that can actually cause brain disorders. Some current research seems to believe that prion or prion-related chemicals, misfolded proteins, are what cause Alzheimer's and other forms of mental dementia. But an even bigger problem about prions is we've learned that if you eat prions from another organism, you can actually get prion-related diseases. Chief example is what you would know as mad cow disease, which is technically called variant Crutchfield's-Jacobs disease, where prions collect inside the nervous tissue in your brain and start to cause it to destroy itself and decay, which causes holes to form in your brain structure, which is why we call it bovine spongiform encephalopathy. Bovine because it's caused by cows, spongiform because it causes holes, and encephalopathy because it's in your brain. In addition to the chaperone and the folding of the protein, a couple other things can happen post-translational. Amino acids can be removed, added to the ends to just finalize the structure, and ultimately multiple polypeptide chains can combine into bigger proteins, and we call that the quaternary, or the fourth level of protein structure. All right, guys, that's it for translation. This one was a mouthful.